What would you say if you heard that our DNA has been altered or seriously messed with? Would you shake your fist and blame the Illuminati or the deep state? Well, unlike most things that can be blamed on them, this one can't. This is something that appears to have happened around 200,000 or 300,000 years ago, so unclench your fist and relax, and I'll try to make sense of this situation. Who knows, maybe we can still find a way to blame this on the Rothschilds. But seriously, you have probably heard the phrase that we only use around 10% of our brain, or some variation of that statement. But what you don't hear is an explanation as to why that is. Science is beginning to figure out why. Our DNA commands the functions of our body. In particular, the second chromosome, the one that is in question, affects our brain. The second chromosome has been altered or modified. This obviously raises a multitude of questions. Who could have done this? What did this change? Why and when did this happen? It wasn't that long ago that scientists claimed that a great deal of our DNA was just junk DNA. This was the explanation they offered for what they didn't yet understand. We now know that was a total fabrication. There is no junk DNA, in fact. There's evidence that some of our DNA has been removed, or even that some has been added, and this does not appear to be the result of evolution. This was a deliberate cutting and splicing that was artificially done. In fact, it is such a sloppy modification that it rules out any theory that this was an act of God, because God surely would have done a better job than this. So, we will try to make sense of what all this means. There's also proof of modifications to our seventh chromosome, and it's responsible for the complexity of our speech. Please subscribe to my YouTube channel if you haven't already, because some of the things I refer to in this video are covered more in depth in other videos I have on my channel. You can watch those to learn more about those topics. Thank you. In the late 1970s, a revolution was brewing in the world of genetics. Scientists, armed with new tools and techniques, were beginning to unlock the most fundamental secrets of our genetic code, peering into the very essence of life itself. The air in the labs crackled with anticipation. A key part of this endeavor involved carefully comparing the chromosomes of humans and our closest relatives, the great apes. Researchers meticulously prepared slides, staining and examining these microscopic structures. Chimpanzees, our nearest cousins, gorillas, majestic and powerful, and orangutans with their distinctive reddish hair all possessed 48 chromosomes, organized into 24 pairs within each cell. This was the established order, the expected norm. Given the overwhelming genetic similarity between humans and chimpanzees, the prevailing expectation was that humans would match that number, neatly slotting into the established chromosomal pattern. It seemed like a logical assumption. But as scientists peered through their microscopes, meticulously counting and recounting, the count for humans stubbornly remained different, 46 chromosomes. A seemingly small discrepancy, but one that held profound implications. This wasn't a trivial difference. It wasn't a mere counting error or a lab anomaly. It was a profound evolutionary puzzle, a chromosomal conundrum that challenged the very foundations of our understanding of human origins. Where did our missing pair go? Had we somehow lost genetic material along the way? Had they vanished without a trace? Or was something else entirely at play? Some hidden mechanism that could account for this numerical disparity? The discrepancy challenged the widely accepted idea of a relatively recent common ancestor, shared between humans and the great apes. It threw a wrench into the established evolutionary narrative. Skeptics of evolution, always eager to find fault lines in the theory, pointed to this chromosomal difference as a significant gap in the story, a potential challenge to the entire framework. How could a species, particularly one as complex as humans, lose an entire chromosome pair and not only survive but thrive? It seemed biologically implausible. The answer, as it turned out, wasn't loss at all, but fusion, a process of chromosomal merging. Scientists began to suspect, driven by mounting evidence, that two ancestral ape chromosomes had, at some point in our evolutionary past, undergone a remarkable transformation. They had fused together, end to end. This fusion event resulted in the formation of a single, larger human chromosome, a composite structure bearing the genetic legacy of its two ancestral components. This elegant hypothesis would neatly explain the numerical discrepancy, resolving the chromosomal puzzle. 24 pairs in apes, the ancestral state, but in humans, two chromosomes fused into one, a unique event in our lineage, ultimately leaving 23 pairs, a testament to this ancient chromosomal union. The search for concrete evidence to support this fusion hypothesis began in earnest, not just counting chromosomes under a microscope, but delving deep into their intricate structure, mapping the precise arrangement of genes and other genetic markers. Researchers became genetic detectives, meticulously piecing together the clues hidden within our DNA, carefully mapping the complex landscape of our genetic code, searching for the telltale signs of this ancient fusion event. They hypothesized that our missing chromosomes weren't truly missing, but rather their genetic information was still present, just joined together in a novel configuration. If true, this fusion would represent a defining event in our evolutionary history, a pivotal moment that set us on a distinct path. The mystery of the missing chromosomes, therefore, became a central question in the field of human genetics, driving research and sparking intense debate. The answer, when it finally arrived, would reveal not just a numerical solution to a chromosomal puzzle, but a profound story of monumental change, a dramatic shift in our genetic makeup that shaped the course of human evolution. I'm sure you are familiar with the double helix structure of your DNA, but what we need to envision is the chromosome. Picture a test tub filled with the genetic material, and on each end a red cap that holds the material inside the test tube. These red caps are called telomeres. Where the chromosome connects in the center is referred to as the centromere. The telomere of one chromosome connects to the telomere of the next chromosome, but in the case of our second chromosome, a centromere is connected to a telomere, or end cap. This doesn't occur anywhere else in our DNA, or in any DNA anywhere else. This is one of the most dramatic and compelling discoveries in the history of evolutionary biology, a revelation that would forever alter our understanding of ourselves. And it all began with a simple, seemingly perplexing count, a numerical anomaly that sparked a scientific quest to unravel one of evolution's most intriguing mysteries, and ended with scientists scratching their head and wondering how this could happen. I know what you are thinking, this sounds like the script to a movie, but I assure you it is not. Our very DNA has been altered, and this is the proof of it. But this isn't something that we have the ability to do. We have technology called CRISPR-Cas9, but that's a different topic for a different video, and it wouldn't produce these results. Within each of our cells lies a meticulously organized library of genetic information. This library isn't a chaotic jumble. Instead, it's carefully arranged into structures called chromosomes. Chromosomes are the organized volumes in the library of you, tightly coiled DNA packed with genes. These genes are the blueprints, the instructions that dictate everything from the color of your eyes to the intricate workings of your immune system. Humans have 23 pairs, inheriting one set from each parent. This dual inheritance ensures genetic diversity and contributes to the unique tapestry of traits that define each individual. Our focus, chromosome 2 and chromosome 7. These two chromosomes, while just a small part of the overall picture, hold fascinating stories within their DNA sequences. Chromosome 2 is a giant, the second largest, a veritable tome in our genetic library. With over 1,300 genes, many crucial for brain development. These genes play a vital role in shaping the complex structures and functions of our brains, influencing everything from cognitive abilities to emotional responses. Its size and content make it a cornerstone of our genetic identity. It's a testament to the complexity of the human genome and the intricate interplay of genes that make us who we are. Chromosome 7, though smaller, is home to the FOXP2 gene, vital for speech and language. This gene has been the subject of intense research, offering insights into the evolution of human communication. FOXP2 doesn't give us language alone, but it's key for the neural wiring that enables articulate speech. It's a critical component in the intricate network of brain regions that work together to produce the sounds and structures of language. Chromosome 2 tells a story of dramatic fusion, a glimpse into our distant past. Chromosome 7, of subtle genetic fine-tuning, a testament to the power of natural selection. Together they offer two windows into what makes us human. They provide valuable clues about our origins, our capabilities, and our potential. One is about architecture, the other about communication. They represent two fundamental aspects of our being, our physical structure and our ability to connect with one another. Both are essential chapters in our evolutionary journey. They highlight the remarkable processes that have shaped us over millennia, leading to the complex and fascinating species we are today. The fusion hypothesis, while compelling, needed concrete proof. It wasn't enough to simply propose the idea. We needed irrefutable physical evidence embedded within our very DNA to validate this extraordinary claim. The scientific community demanded tangible data, something that could be observed and verified. Normal, unfused chromosomes possess a single critical structure known as a centromere. This specialized region plays a vital role in cell division, ensuring that each daughter cell receives the correct number of chromosomes. Furthermore, they also have protective caps called telomeres at each end. These telomeres safeguard the integrity of the chromosome, preventing degradation and maintaining its stability over time. They are essential for the long-term health and functionality of our genetic material. Now, if human chromosome 2 was indeed the result of a fusion event, a merging of two ancestral chromosomes, it should logically exhibit telltale signs of this union. Specifically, it should possess two centromeres, a relic of its dual origins. Moreover, we would expect to find telomere sequences not just at the ends, but also buried within the middle of the chromosome, at the precise point where the fusion occurred. These internal telomere sequences would serve as a genetic signature of the chromosome's past. Driven by this hypothesis, scientists embarked on a quest to find these genetic scars, these remnants of a distant evolutionary event. The search was on for definitive proof that chromosome 2 was indeed a fusion product. In the 1990s, the advent of advanced DNA sequencing technologies revolutionized the field of genetics. These powerful tools provided scientists with the ability to decipher the intricate code of life with unprecedented precision. This breakthrough allowed researchers to zoom in on chromosome 2, scrutinizing its structure at the molecular level. They could now examine the chromosome in minute detail, searching for the elusive evidence of fusion. And indeed, they found telomeric DNA. The sequence is normally found exclusively at the tips of chromosomes, acting as protective caps. But this time, these sequences were buried deep within the center of chromosome 2, exactly where the fusion was predicted to have occurred. This was a major breakthrough, a key piece of the puzzle. Further analysis revealed that these telomeric sequences were arranged in a head-to-head configuration, precisely as one would expect if two chromosome tips had been joined together in a fusion event. This head-to-head arrangement provided further compelling evidence supporting the fusion hypothesis. This was the molecular seam, the point where two chromosomes had once been joined, leaving behind a telltale scar in our DNA. This discovery was the smoking gun, the definitive proof that chromosome 2 was indeed a fusion of two ancestral chromosomes. Adding to the evidence, they also found a second, inactive centromere, a non-functional remnant of the chromosome's dual ancestry. This inactive centromere served as a genetic fossil, a vestigial structure providing further insight into the chromosome's evolutionary history. The case for chromosome fusion was now irrefutable. It was clear that human chromosome 2 was born from the union of two distinct ancestral ape chromosomes, a pivotal event in our evolutionary history. With all the evidence they found, there was still no explanation of why, or how that this occurred. This fusion event was not a common occurrence. It was a singular, unique event that transpired specifically within our lineage, setting us on a distinct evolutionary path. It was this fusion that ultimately set us apart from our closest relatives, contributing to the genetic divergence that defines our species. The story of our origins was now indelibly written, not just as a theoretical concept, but as a tangible reality encoded in the very fabric of our cells, in the intricate architecture of our chromosomes. The long-standing mystery of the missing chromosomes, the genetic puzzle that had intrigued scientists for years, was still incomplete, revealing a profound insight into our evolutionary past. The fused telomeres in chromosome 2 are a powerful testament to human evolution. Or is it evidence of manipulation? Scientists, as I'm sure you guessed, disagree on this. This scar from our past raises more questions than it provided answers. It's a universal feature found in every human, linking us to a single pivotal event. The theory of common descent does not explain this fusion, and it threatens the theory of evolution presented by Darwin. This is how science works: observation, hypothesis, prediction, and confirmation. The fusion is a relic, like the appendix or vestigial leg bones in whales. It's a story of our shared ancestry, written in our DNA. But who is the author? It leaves a lot of room for doubt. I'm reminded of the story of Enlil and Enki. They were said to have had a disagreement on just how smart humans should be allowed to be. The verdict, maybe after they modified our DNA, possibly they decided to go back and make a few adjustments. Had we become too smart? Too hard to control? I think the evidence says there was something that appears to have happened. and it isn't the result of evolution. If you want to learn more about the story of Enlil and Enki, you can watch the video I made about them. I also have videos that cover human consciousness that might help you understand more of this. The fusion event created a reproductive barrier. 46 chromosome humans couldn't easily breed with 48 chromosome relatives. This isolation was a genetic fork in the road, setting our ancestors on a separate evolutionary path. In apes, 2 medium-sized chromosomes, 2A and 2B, match the 2 arms of our chromosome 2. It's like taping 2 pages together to make a new chapter, or like ripping the end of chapter 2 out of a book and combining it with the 3rd chapter. You will notice the page is missing, but in this case, it was a little more well hidden. The fusion likely happened after our lineage split from chimpanzees 6 to 8 million years ago. It marked the birth of the human line, but some scientists think it was much more recently. Chromosome 7's FOXP2 gene tells a story of subtle change with huge impact. The KE family in Britain, with a mutation in FOXP2, struggled with speech and grammar. This was the first gene directly linked to a human language disorder. FOXP2 isn't the language gene, but it's a master switch for neural circuits needed for speech. The human version of FOXP2 has 2 unique changes not found in other primates. These tweaks likely enhanced our vocal learning and motor control for speech. The changes appeared around the time modern humans emerged. This gave us a massive advantage: complex communication and culture. A small genetic change, a giant leap for humanity. Chromosome 7's story is about the fine-tuning that made us truly human. Chromosomal fusion is risky, often harmful, sometimes fatal. For the fusion to persist, it must have been harmless or even beneficial. One theory: the fusion brought key genes closer, changing how they're regulated, possibly boosting brain development. Another: the fusion created reproductive isolation, locking in other advantageous traits. Or maybe it was a random event that spread by chance in a small population. Once established, it set the stage for further human evolution. We are told the truth is likely a mix of luck and advantage, a single event with far-reaching consequences. Our chromosomes and our brains are deeply linked. The human brain is 3 times larger than a chimp's, with a massive cerebral cortex. Chromosome 2's fusion may have catalyzed this brain expansion. Genes like ASPM on chromosome 2 regulate brain size. The fusion could have unleashed their potential. FOXP2 on chromosome 7 gave us the vocal control to use our big brains for language. Communication allowed for culture, cooperation, and shared knowledge. Evolution often repurposes old genes in new ways. Chromosome 2 was a major renovation, FOXP2 a crucial upgrade. Together they built the modern human mind. The changes to chromosomes 2 and 7 are more than curiosities, they're markers of our identity. The fusion of chromosome 2 is a molecular scar uniting all humans. FOXP2's tweaks helped spark our unique communication. These events are clear, profound milestones in our story. Our DNA is a living record, connecting us to our ancestors and each other. The tale of our chromosomes is a story of connection across time, across humanity. For all our differences, we are one species, shaped by extraordinary evolutionary history. And as we keep decoding our genome, the story of how we became human continues to unfold. Were we modified to limit our potential, to limit our spirituality? I don't know the answer to that, but it's very suspicious. Thank you for watching, and please subscribe. Also, I urge you to watch the other videos referenced in this video to help you understand more of this story. And as always, stay informed.